This is Microbiology Lecture 10. What we're going to do is look at prokaryotes. Remember that there are two domains in the prokaryotes, bacteria and archaea. This lecture has two slides at the end on archaea. All of the rest of it is about bacteria. So we don't really consider archaea too much since there are no members of the domain archaea that can act as pathogens in humans. None yet discovered, at least. Okay, so just as we did with originally with the viruses, and then in the last lecture, I believe we we did with the helminths and algae and fungi and protists. We're going to go through just a few basic characteristics and then talk about individual members of the bacteria, introducing the names and just a few things about each one. We'll show how they're characterized or classified, but I wouldn't worry about classification. I'll explain that in a minute. So, as I said, there are two domains, bacteria and archaea. Characteristics of these include one large circular chromosome. There are no histones associated with the DNA. There are no membrane-bound organelles inside of the organisms. There is peptidoglycan in the cell wall of bacteria, but not in the cell wall of archaea. And archaea and bacteria both divide by binary fission. Now the domain bacteria, using a technique called polymerase chain reaction, or PCR, there is strong evidence that there can be as many as 10,000 different bacterial species. It should be in here. There can be as many as 10,000 different bacterial species per gram of soil. Of course, the number of potential bacteria per gram of soil, actual individual bacteria per gram of soil, is in the trillions or more, but 10,000 different species is very likely to be found in an ordinary gram of soil out there. Many of the bacteria, in fact, have not been identified and not been characterized because they can't be cultured. They haven't been cultured out of the samples taken from the environment. Often it's because they have special nutrient needs. Quite often there are bacteria out there that can only grow on the metabolic byproducts of another bacterium. So their nutritional requirements are extremely complicated. There is a complex food chain present in the environment, requiring products from one bacteria in order for another to grow. And if we can't culture bacteria, then it's very difficult for us to identify it or understand its role, ecological role, or its metabolism. Okay, there are several different phyla in this domain of bacteria. The different classes within this particular phylum, the protobacteria, are designated by Greek letters. And on the following slides, I'll show alpha class, beta, gamma, delta, epsilon. All those letters are here for those of you who don't know it. Alpha, beta, gamma, delta, and epsilon. But other than knowing that there are designations by Greek letters for these different classes in this phylum, I wouldn't worry about that. I wouldn't worry which one is an alpha, which one is a beta, a delta, gamma, whatever. Just the fact that they are classified that way. Here you see alpha, but again, don't worry about it. Now, I already told you that that organisms in general are often identified using a dichotomous key, and that's what you see here. And that's enough said about this. I'm not going to get into it. Are there important genera among these bacteria that are pathogenic in humans? Yes. This genus Brucella does cause brucellosis. Enough said. Rickettsia, this genus, and there are several species out there that can cause disease. Rickettsia causes different kinds of typhus, rickettsial typhus, and again, that's it. So there are all kinds of alphas, and they have quite a wide range of potential metabolic characteristics, none of which I'm going to go into at all in any great detail. These are betas. There are some important members in this group of proteobacteria, and here are some key examples. The genus Neisseria has 2 species members that cause disease in humans. One is Neisseria meningitidis. This causes meningococcal meningitis. It's not the only kind of meningitis. Meningitis, of course, is inflammation of the meninges, the linings of the brain. Meningococcal meningitis is caused by Neisseria meningitidis. And there is Neisseria gonorrhea, which causes the sexually transmitted infection, STI. Sexually transmitted infection, gonorrhea. So 2 important species in this genus that you need to know, be familiar with, Neisseria meningitidis and Neisseria gonorrhea. And of course, we'll go through them in greater detail when we look at microorganisms causing disease in the nervous system and microorganisms that cause diseases which are diseases of the genital urinary system. One key characteristic, and I've mentioned this before, of Neisseria is that it typically, this is, in my opinion, a beautiful photomicrograph. I mean, no, it's not a photomicrograph, it's an electron micrograph. It's quite amazing. In any case, these are Neisseria. I don't know if it's gonorrhea or meningitis, but it's growing as in pairs. The arrangement that it grows in are pairs. Very, very, very characteristic feature. Okay. Bordetella, important organism. We're still in the betas, but I wouldn't worry about this being beta or Neisseria being beta or anything else I said. Just know that, yes, Greek letters are used for proteobacteria. Okay. Beta, this member, important genus, Bordetella. One key member that causes disease in humans is Bordetella pertussis. Pertussis is a disease which also goes more commonly by the name whooping cough. Very serious. Luckily, there is a quite effective vaccine against it. That's Bordetella pertussis causing whooping cough. These are gammas. Pseudomonas. Pseudomonas, a very commonly occurring Pseudomonas species member of the genus Pseudomonas is Pseudomonas aeruginosa. It is pretty much everywhere. It's omnipresent, associated with humans on the surface, outside of the humans typically, and it is a pathogen in terms of being an opportunistic pathogen. The reason and as I've explained to you previously, opportunistic pathogens will infect and cause disease in individuals who have some kind of deficit in terms of protective mechanisms or immunosuppressive, or people who are immunosuppressed in some way. It is an important cause. Pseudomonas aeruginosa is an important cause of nosocomial infections. Nosocomial infections are infections that are picked up in the hospital. So in a healthcare facility, there are certain organisms around that are more commonly present, and some individual patients pick them up. So it's an important cause of nosocomial infections, particularly in burn or cystic fibrosis patients. Characteristic infection in the lungs of cystic fibrosis patients is caused by Pseudomonas aeruginosa. One characteristic of it is that when it grows on a petri dish or an agar, or I guess an example of this would be when it causes an ear infection, it releases a green fluid or a green stained metabolite, and so if you see green staining in your culture or a green fluid coming out of someone's ear, odds are it's going to be a Pseudomonas aeruginosa infection. Another important genus is Legionella, and the species that's important in humans that causes disease is Legionella pneumophilia. This is also called, this is also the cause of, and the more common name, and the common name for the disease is Legionnaire's disease. I'll spell that for you, Legionnaire's disease. Actually, let me just make an alteration here. Yes. Legionnaire's disease. It is a respiratory infection picked up by people who breathe in the organism. The organism typically grows in warm water in containers that are sitting around for a long time with warm water, like air conditioning units on the top of buildings, or warm water pipes blowing air. Ventilation systems in buildings can pick it up, have it growing in the system and blow it into the building. An important organism causing this disease, Legionella pneumophilia. Pneumophilia, of course, refers to the fact that it is it is more typically found growing in lungs when it causes disease. Okay. The next organism that I introduce is called Vibrio cholera. Now, Vibrio cholera is the cause of the disease cholera, which is a gastroenteritis, severe gastroenteritis. It used to be the cause of very serious epidemic outbreaks. It would kill large percentages of population when there was an outbreak. It is typically transmitted by a fecal oral route. So if you get contamination of drinking water, which used to be extremely common, then you can have that. Nowadays, typically, at least in most countries, would have safe water drinking. Sometimes, well, in industrialized countries, it's piped right into the house, and it's a safe drinking water. But in other countries, there are available clean drinking water supplies. Typically, outbreaks occur when there is flooding or some kind of natural disaster, earthquakes, things like that, where you get a disruption of the clean water supply, and people start drinking water that's been contaminated in some way. The characteristic description of Vibrio cholera that every student typically learns is that it is a, did I say comma? Vibrio cholera is that it is a comma-shaped organism. It has this characteristic comma shape, this curved shape. There's another one, there's another one, and there's another one. Very commonly, this kind of curved shape. Vibrio cholera used to be very feared, and there were outbreaks, used to be in the 1800s in Montreal, outbreaks until clean water supplies became available. There are an entire family of bacteria which are called the Enterobacteriaceae. So the Enterobacteriaceae are a group of gram-negative enteric bacteria. All of them happen to belong to this grouping. Again, I wouldn't worry about it, what Greek letter it's named by, but all of these are gram-negative enteric bacteria. All the enteric bacteria are of interest and possible. There are species in each of these genera that can cause serious disease, and we will talk about that when we talk about diseases associated with the gastrointestinal system. I would at least become familiar with some of these, at least so you can recognize the names, to realize that these are all gram-negative enteric bacteria within the Enterobacteriaceae family. You know, some of them, like Salmonella, is very commonly known to certain species cause disease. Some members of E. coli, but actually all of them can. They are identified one from the other by culture of fecal samples using setups like this, the Entero tube, which I talked to you about before when we first started, I think the first lecture, where you take a sterile packaged in, they send the fecal sample down to the lab and they take a sterile packaged Entero tube, open it, pull out the sterile sampling metal stick, touch the ends to the fecal sample, and then insert it right into each compartment in the Entero tube. These compartments are separated by wax seals, which when they pull it out, they push it in, pull it out, and the sample is closed. The tube's removed, the sample's closed, and the wax seals around the probe or needle. And each compartment has reagents in it that give a characteristic result, either change in color or gas formation, things like that. So it can readily identify one type of enteric organism from the other. Enteric refers to organisms that grow in the gut, and typically, not all of them, but many are in fact gram-negative enteric. Most enteric organisms are in fact gram-negative bacteria and belong to one of the genera that I just listed on the previous page that are in the family Enterobacteriaceae. Okay. Now another important genus is Pasteurella. There are members that can cause pneumonia and septicemia. Well, these are not examples of Pasteurella. These are other members within that grouping, but Yersinia pestis is the organism that causes bubonic plague. The plague was also a serious cause of terrible epidemics and pandemics all over the world, killing large percentages of the human population in previous centuries. It was also called, the disease was also called the Black Death, or in French, la peste. La peste is, or the plague in English. La peste is the plague in English, is also the name of a very famous book by an author, Albert Camus. Here you see the front cover of a copy of The Plague, so it's in English, this one. Albert Camus, his name is more prominently displayed because he, in fact, is perhaps even more famous than the book. He was a Nobel Prize winning author. A book worth reading, I must say. I read it, and when I first started studying microbiology, it was recommended in the first introductory university course I took. Really quite a phenomenal book. Haemophilus influenza can be the cause of meningitis. This, of course, would be a non-meningococcal meningitis. Meningococcal meningitis is, of course, caused by Neisseria meningitidis. Haemophilus influenza is the cause of non-gonococcal meningitis. Francisella tularensis, I'm not going to talk about this. It does cause this disease, which is normally occurs in people who trap animals for their fur. Okay, we can move on. Quite a bit, actually. We will talk about these two on the next slide. The next two slides. Campylobacter is a genus that has a couple of species. This should be in italics, but so there's Campylobacter jejuni and Campylobacter coli that are microaerophiles. I've talked to you about those before. These are organisms which require oxygen, but at a lower level typically than what is found but at a lower level than what is found typically at in the atmosphere. And so they're called microaerophiles, and they live in the gut. It says rickettsia here, which is an error on this slide. I shouldn't show this diagram. They cause a gastroenteritis. The next slide shows Helicobacter, also a microaerophile. Helicobacter is an interesting organism in that the species that infects humans is called Helicobacter pylori. It causes peptic ulcers, first gastritis, and that can sometimes progress to peptic ulcers, and then it can also from there progress to actually stomach cancer. So, and this was actually declared to be a cancer-causing bacterium, the only known cancer-causing bacteria. There's plenty of viruses that can cause formation of tumors, but no other bacteria that's known to lead to a cancer. In this case, it's a stomach carcinoma of the epithelium. It had an interesting discovery. Peptic ulcers were fairly I'm sorry, okay. It had an interesting discovery back in the association between this particular bacteria and gastritis and peptic ulcers has an interesting backstory. A Australian physician scientist was quite convinced, based on his observations of patients with peptic ulcers and examination of the tissues, that this bacteria was the cause of peptic ulcers. And he was getting absolutely no traction, no matter how much he presented his results, presented the data. Everyone was had completely accepted and was resistant to anything other than the idea that a abnormal secretion of stomach acid, or hydrochloric acid in the stomach, was the cause of peptic ulcers. They wouldn't accept it, and he actually, some might say it wasn't wise, but it did work. He drank a culture of this bacteria and almost immediately developed gastritis. And soon after treatment with self-treatment with antibiotics, it was cleared. The infection disappeared and the gastritis disappeared. And so people started finally actually listening to him and looking at his data and looking at it objectively. It was actually a big enough discovery that he was awarded the Nobel Prize, because it prevented development of peptic ulcers in thousands and thousands of people. And peptic ulcers can be quite debilitating and painful and damaging, and also prevented many cancers developing, stomach cancer. It has a classic kind of shape and has flagella at one end, Helicobacter pylori. Okay. Let's move on. So those were protobacteria, which are, classified according by groups with Greek letters, but I didn't require you to know which one was in which group. Just that we just covered the names of several important pathogenic bacteria. Now let's look at non-proteobacteria. These are gram-negatives that are non-proteobacteria. This includes in this group cyanobacterium. Cyanobacteria are an unusual group that use light as an energy source. They're the only oxygen-producing photosynthetic bacteria. They were, in fact, originally thought to be an algae, and are still called blue-green algae. Most commonly called blue-green algae, but it's in fact a bacteria. And it does produce, some strains do produce toxins, which can be quite deadly, particularly for some animals, certain animals more than others. Here you see the cyanobacteria. They look like algae, certainly, and I can understand why for years and years. I mean, it was only recently that they realized they were bacteria. Okay, there are also gram-positives. The gram-positive bacteria are divided typically on the amount of G plus C content in their DNA. There are two major groups, low GC and high GC. Some organisms are low GC. We'll talk about those. Again, I would never ask you if it's a low GC or high GC. These are all gram-positive organisms. This is an especially important group that I've spoken to you about before. This particular genus, Clostridium. All members of the genus Clostridium that we talk about are obligate anaerobes. They will not be able to live in the presence of oxygen. Oxygen is toxic to them. In fact, in some cases, they even use oxygen as a treatment on a wound that is infected with this organism here to try and kill the organism off. These are obligate anaerobes that also produce endospores. I've spoken to you before about endospores. Here you see a culture of bacteria that have just formed endospores. You know it's recent because the endospore is present here. You see an endospore, and here you see an endospore inside the bacterium. The vegetative part of the organism and the spore coat forms around the chromosome, preserving the chromosome, and it can preserve it for a long time. The vegetative part of the organism here, all of this, will soon break down and break away, and you'll just be left with the endospores, which can remain in a suspended state for hundreds of years, if not more. Nobody knows exactly how long. The key members that are important, or species that are important, are Clostridium tetani, which causes the disease tetanus, Clostridium botulinum, which causes botulism, Clostridium perfringens, which causes gas gangrene, and Clostridium difficile, which causes a diarrhea and colitis. I'm going to add that in. Colitis. Nope, not that way. I'm going to add it in like this. Colitis, which is, of course, inflammation of the colon, large intestine, or colon. Colitis. There is also Bacillus, and an important member of this genus is Bacillus anthracis. Bacillus also produces endospores, and in the lab we typically would carry out experiments using a non-pathogenic species member called Bacillus subtilis, very, very commonly used in the lab. Bacillus subtilis, non-pathogenic. The pathogenic form, one pathogenic species is Bacillus anthracis, which causes the disease anthrax. Okay. Now, Staphylococcus. We've spoken certainly about Staphylococcus aureus before. I've shown you photomicrographs and talked about it. There are many different strains, subspecies strains. Okay, this is of course the genus. There's the genus right there. Staphylococcus and aureus is the species, but there are many substrains of it. Some are normal microflora, but others are pathogenic. Everyone has this on their skin or inside them, but there are some that are pathogenic. They can cause infections and surgical wounds, and they can also cause quite a serious disease process called toxic shock syndrome. They are sometimes responsible for food poisoning, and of course the one that people are familiar with is the methicillin resistant Staphylococcus aureus, or MRSA, which can cause problems and infections, particularly because it cannot be treated with methicillin. Now, there is a genus. It is not a pathogen, but it is very commonly used for food production, and so I think you should be aware of it. This bacterial genus is Lactobacillus. There are several different species, all used in food production, and the kinds of foods that are produced using Lactobacillus include, cheeses and yogurts and things like that, dairy products that are fermented. There is also Streptococcus, and please do not, one problem with Staphylococcus and Streptococcus is that because they start with an S and all you have sometimes is the S period, people mix them up. So Staphylococcus aureus, that's separate. Now we're talking about Streptococcus, and an important pathogenic species of Streptococcus is Streptococcus pyogenes. This is also called Group A Streptococcus, or gas organisms, GAS standing for Group A Streptococcus. It is a pathogen. The gas organisms are pathogens. They can cause a number of different disease processes, including scarlet fever, pharyngitis or strep throat, also sometimes called streptococcal pharyngitis, rheumatic fever, erysipelas, and impetigo. A lot of young kids, little kids catch, again, impetigo. It's a crusty infection around the mouth. They stick all kinds of stuff in their mouth. Every little kid does. And this is caused by Streptococcus pyogenes. This is probably one of the most harmless compared to of all these. I mean, some of these are very serious. Another, I'm sorry. Another important species is Streptococcus pneumoniae, which causes pneumonia. We spoke about Streptococcus pneumoniae when we were talking about capsules and transformation of bacteria, transformation by genes coding for capsules, so the transfer of naked DNA between bacteria. And I mentioned to you that the encapsulated form, the form that has a capsule, so encapsulated form, is more pathogenic because the capsule inhibits phagocytosis by phagocytic cells like macrophages or neutrophils, and so this and the ones with a capsule are more pathogenic. This is not cheese, but let's get rid of this. This is a photograph of a patient's throat. This patient has strep throat caused by Group A Streptococcus or Streptococcus pyogenes, same thing. So they have strep throat, which is, it feels, I've had it, it feels like your throat is on fire. It really feels bad, especially when you swallow. I believe there's another photo. Yes, that's also equally ugly. Okay. There are also organisms that belong to the genus Enterococcus that cause disease. Several species frequently the cause of hospital infections, which are, of course, also called nosocomial infections. They have a high rate of antibiotic resistance, so often it's difficult to treat, especially if it's developed vancomycin resistance. So there are VRE organisms or vancomycin resistant Enterococcus. It's a problem. Another organism is Listeria monocytogenes, which causes the disease listeriosis. Okay, we did talk about Streptococcus pneumoniae causing pneumonia. There is another organism, bacteria, again, these are all bacteria that we're talking about, which causes a milder form of pneumonia. It's called Mycoplasma pneumonia. The common name, some people call it walking pneumonia. It's a milder pneumonia. It's called walking pneumonia because people aren't laid up in bed. They're functioning, but they're sick, and they have real trouble shaking this off. Okay, so those were low GC content gram-positive. These are high GC content. Corynebacteria is an important genus because it has one species member, Corynebacterium diphtheria, which causes the disease diphtheria. Again, we have a vaccine against this. Excellent. Serious disease. Then there are the Mycobacterium organisms. This genus has 2 members that cause disease. I've spoken to you about them before. These are acid-fast organisms. They will resist decolorization in an acid-fast staining procedure, decolorization by acid alcohol. So they're acid-fast or acid-resistant, and that's how they're identified. Mycobacterium tuberculosis is an important species member of the genus. It causes the disease tuberculosis or TB, and Mycobacterium leprae, which causes leprosy. There are, I think, I gave you some numbers. I think it was something like 1 million new patients per year worldwide or more. I can't remember. Something huge, just gigantic, of TB. In the lab, we use a non-pathogenic organism for acid-fast staining. It's called Mycoplasma smegmatis. There is also an organism which does not cause any serious infections. Typically, it is called Propionibacterium acnes. This is the genus Propionibacterium. It is found on the skin of normally on the skin of virtually every person ever sampled in the world, anywhere. Let's move on. And these are assorted groups, different groups of organisms. We're almost at the end of this survey. It's essentially just a survey, introduction of each important pathogenic bacteria that cause important diseases, that are things that are infectious, and you will come across certainly quite a few of these in the hospital. Chlamydia trachomatai causes eye infections, and that kind of disease process leads to the disease trachoma. It's associated with many cases of blindness throughout the world. It is also a sexually transmitted infection, or STI, causing lesions on the genitalia. Trachoma, the disease that causes blindness, there are approximately 84 million active infections each year. It can be treated as a sexually transmitted disease. There are approximately 127 million per year. These are statistics, I believe, the most recent available ones from 2016. So, we're talking in terms of disease process in the eye leading to blindness. Not that they all become blind, but many do. Most are treated and overcome the infection, but many are not properly treated and end up blind, which is a tragedy. 84 million cases of trachoma and 127 million cases of sexually transmitted disease, all caused by Chlamydia trachomatai. It causes, in terms of sexually transmitted infection, it causes non-gonococcal urethritis. What that means is that it is not caused by Neisseria gonorrhea, so non-gonococcal urethritis. Neisseria gonorrhea causes the disease gonorrhea, which is a sexually transmitted infection and does cause urethritis. This causes non-gonococcal urethritis. Another organism, and this is the third one we've talked about here now, that causes pneumonia. The first one was Streptococcus pneumonia. The second one was Mycoplasma pneumonia, and this is Chlamydia pneumoniae, which can also cause pneumonia. This last one is, it's a disease in parrots. Your pet parrot could give this to you, psittacosis. Let's not talk any more about it. I've given you links here in this for trachoma, from the World Health Organization, and sexually transmitted non-gonococcal urethritis, a link to the CDC, giving you quite a bit of information about these. I am not going to speak about this slide or this one. This is an important organism. The genus is Treponema. The species member is Treponema pallidum. It is the cause of syphilis, a sexually transmitted infection. So far, we've talked about several organisms that cause sexually transmitted infections, including Neisseria gonorrhea, non-gonococcal urethritis caused by chlamydia trachomatai, and here Treponema pallidum, which causes syphilis. So, you know, that's a huge number of infections, actually, of sexually transmitted. The majority, I would guess, of sexually transmitted infections fall under those, you know, gonorrhea, chlamydia, non-gonococcal urethritis, and syphilis. That covers a lot of territory. Let's not talk about bacteroides. It's there, but we'll move on. Now we get to the last two slides, and the last two slides are about the domain Archaea, which is also a protozoa, not a protozoa, which is also a prokaryote. Bacteria, the domain bacteria and the domain Archaea are two domains, the two domains in the prokaryotes. The third domain, of course, is eukarya, the eukaryotes. They live in extreme environments. Some are hypothermophiles. They can live at extremely high temperatures. This is under high pressure, deep in the ocean, so that the water's not boiling because it's under such high pressure. But the organisms live there. Methanogens, which produce methane, or extreme halophiles, which are organisms that can grow at a very, really high salt concentration, hypertonic surroundings. And they're fine. Let's move on, and I'm not going to talk about this. And so that's the end of Unit